Hello and welcome everyone to this, our second PACE webinar, where we would like to introduce you to the PACE programme and to provide information about our second funding round, which is currently now open. I am Dr. Bev Isherwood. I'm one of the PACE programme directors at PACE, and I'm joined on the webinar today with a number of the PACE team. We are running this webinar to support the new PACE open call for Diagnostic Innovations with the potential to help address AMR. In today's webinar, we'll start with a brief background and introduction to PACE, who we are, what we do, and that will be presented by my colleague and PACE co-director, Dr. Clive Mason. And then following Clive, Dr. Pete Coombs and Dr. Tanya Taylor, two of our PACE scientific leads, will provide an overview of the open funding call with the key features, including the scope, eligibility, the application process, timelines, selection criteria, and also share information about the resources available and what we hope as PACE to be able to offer applicants and successful applicants. Before we go into that, just a bit of housekeeping. So as this is a webinar, we can't see or hear you. So should you need any technical assistance during the webinar, please use either the chat function or the Q&A function, and someone will be able to help. We would also very much welcome you to submit any questions you have in the Q&A box throughout the webinar. We will aim to answer these questions towards the end of the session, when we will have an interactive Q&A session with the speakers alongside additional members of the PACE team. A recording of this webinar and a PDF of the slides, plus a full set of frequently asked questions, will be made available on the PACE website shortly after this webinar for you. I think that's all the housekeeping I need to share. So without further ado, I am going to hand over to Clive at this point to introduce you to PACE.
My name is Clive Mason, and alongside Bev, I am a Programme Director for the PACE initiative. I am an employee of LifeArc. It is my pleasure to briefly set the scene and introduce the ambition and intent for PACE and this new funding call. PACE is a UK-based £30 million programme of funding and support to be deployed over 5 years. PACE will bring together the right funding, resources and partnerships to help innovators progress their early-stage antimicrobial drug and diagnostic projects with greater speed, support and confidence, giving the best AMR innovators the greatest chance of success. PACE is a collaboration between 3 leaders in the UK's health innovation and research community: LifeArc, Innovate UK and the Medicines Discovery Catapult. PACE harnesses their unique expertise, catalysing and working with the global AMR community to accelerate the speed of innovation to mitigate the risk of AMR. As we know, AMR poses a real threat to healthcare systems. Diagnostic testing has the potential to have a significant impact in mitigating the risk and spread of AMR. Currently, the majority of antibiotic prescribing in primary care is empirical. Implementation of diagnostic testing can support antimicrobial stewardship and reduce inappropriate use of antibiotics, can improve patient outcomes, and fully enable the clinical use of new, highly selective antimicrobial therapeutics. We recognise that AMR diagnostics is a massively underfunded area of research. PACE is looking to support innovative AMR diagnostic approaches, sitting alongside and complementing other funding mechanisms that exist in this area. There are 3 main strategic pillars to this PACE initiative. 1, strengthening the pipeline. We want to grow a pipeline of high quality antimicrobial drugs and associated diagnostics aligned with unmet needs. 2, enable preclinical translation. PACE will provide innovative researchers in academia and SMEs the funding, advice and support they need to build de-risked and investment-ready assets. 3, accelerate progress and overcome barriers. We aim to convene collaborative networks to identify barriers to innovation, advance programs and drive new collaborative R&D. Through these mechanisms, we want to further embrace innovation and maximize the diversity of approaches that look to meet the threat posed by AMR. Through the PACE initiative, we will look to provide wraparound support for AMR products and their developers, going beyond a simple funding model. We aim to deepen and strengthen the data packages for AMR assets and work alongside product developers to identify the best partners to plug gaps in resources or capabilities. The intent is to robustly move assets forward to a position that improves the potential for future onward progression and investment. The AMR research community is small, and we want to ensure stakeholders are joined up and aware of each other and their capabilities. We want to ensure we apply best practice and lessons learned. This will bring increased opportunities for collaboration across the AMR ecosystem and identify additional stakeholders that can contribute, shape projects and address barriers. Where appropriate, we will also look to establish and offer enabling capabilities that have the potential to deliver cross-project impact. The aim and aspiration for PACE is to fully enable early translational science onto innovative AMR assets, achieving this through funding, but importantly also through additional wraparound support that maximises the potential of the assets and improves the likelihood for further onward progression and investment. The AMR clock is ticking and the pipeline of AMR-focused products desperately needs to be strengthened. Thank you for your attention.
Hello everyone, I'm Peter Coombs, a member of the PACE team and Scientific Director at LifeArc. Today, I'll provide you with an overview and key features for our new PACE funding call around diagnostic innovation. Please submit any questions you have and we'll endeavour to address these in the Q&A session at the end of the webinar. We are looking to help create and advance an exciting and diverse pipeline of early stage in vitro diagnostics aimed at improving diagnosis and management of bacterial infections with high unmet need. For this funding call, up to £5 million is available to support up to 8 projects. We aim to provide participants with the funding and support that generate key data to allow onward development. The portfolio will be actively managed with projects receiving support and advice from the PACE team. We are focused on technologies that are going to help tackle AMR specifically through innovation and diagnostics for this call. The scope of this funding call is focused on relatively early stages in diagnostic development, around TRL 3 to 5. We'll get back to what we mean by that exactly later on. But at the top level, we're looking at the development of in vitro diagnostics through proof of concept or technical feasibility into early prototype stages, including field studies for generating data in the relevant environment. More details on this later in the webinar. We're happy to consider late-stage TRL2 projects that have particularly strong rationale and supporting data, and a bit more details on that later on as well. We're looking for projects addressing unmet diagnostic needs in bacterial infections with the highest burden of antimicrobial resistance, with the aim to reduce inappropriate antibiotic prescriptions, provide faster results that indicate which antibiotics should be used, and catalyse the move to personalised narrow-spectrum treatments. For this call, we are prioritising projects that are developing diagnostic tests for indications of highest burden, and for which PACE is also supporting the development of novel antimicrobials. Indications in scope are urinary tract infections, lower respiratory tract infections, or bloodstream infections or sepsis. For UTIs, we are seeking rapid, near, or point-of-care diagnostic tests that can discriminate between bacterial, viral, and non-infectious syndromes and/or can identify the most appropriate treatment compatible with use in primary care. Innovative solutions for use at home or in the community setting are encouraged. For lower respiratory tract infections, we are seeking rapid, near, or point-of-care diagnostic tests for RTIs that can discriminate between bacterial, viral, and non-infectious syndromes applicable to primary, community, and hospital triage settings. We're also seeking rapid point-of-care tests that identify the most appropriate treatment, and ideally the underlying pathogens applicable to primary care or hospital settings. Minimally invasive or burdensome sample types will be prioritised. For BSI sepsis, we are seeking innovative methods of rapid pathogen identification and antimicrobial susceptibility profile for suspected bloodstream infection or sepsis cases direct from sample, including for detection and management of the bacterial component of neonatal sepsis. There are more details with preferred characteristics and examples which will be made available to everyone alongside this seminar. So what kind of projects are we looking for? As well as novel technologies, we're interested in projects that include adaptation of existing diagnostics that could be implemented in new healthcare settings or areas, or deployed for a new use case. We would welcome proposals that include potential for community care or at-home use. In terms of biomarkers, both bacterial and host-based or a combination is fine, and we're particularly keen on applications that provide tailored solutions to resource-limited settings, including in LMICs, and aim to keep the cost of diagnostic down as much as possible. Our aim is to support diagnostic innovation with the greatest potential for impact. To help with clarity, I'd like to take you through some of what is out of scope. Firstly, any work beyond TRL5, so later stage diagnostic development, TRL6+, so significant size clinical trials. We are not looking for diagnostics that require central lab infrastructure and high levels of technical expertise or training to use. We're not looking for standalone pathogen ID, standalone technology method or device development. It needs to be a product development project for a specific diagnostic for bacterial infections. Similarly, standalone algorithm or machine learning model development, though incorporation of such things into an IVD project would be fine. We are not supporting standalone biomarker identification or validation or pilot lot production of reagents or instruments. We are not looking to support surveillance-only approaches, nor diagnostics solely focused on fungal, viral, parasitic infections, STIs, or TB for this call. Projects over 2 years in duration also not in scope. So this slide provides further guidance on what we mean by the different TRL levels to help determine your entry project stage, as well as provide guidance on what activities we'd expect to see in the project plan. So for early stages, we expect an outline of the intended use, including an understanding of the competitor market, as well as demonstration of the concept on contrived samples. At TRL3, we're additionally expecting a clear statement of intended use and design inputs defined, as well as lab-based demonstration of the end-to-end process and proof of concept on clinical specimens. At TRL4 to 5, we additionally expect design outputs for a minimal viable product to be set and a regulatory strategy in place, as well as performance data on real samples in lab settings and proof of concept in the clinical setting. To outline the key eligibility for the funding call, we welcome applicants globally. These can be academics or researchers from SMEs worldwide. This can involve consortia, and just to note that large diagnostic industry partners would be permitted, but must cover their own costs. The application should come from a single lead applicant. You can have co-applicants, but this is not a requirement of the funding call, and we accept proposals looking for match funding. The project itself should be planned to last up to a maximum of 2 years, and will be required to be milestone driven with clear go, no-go progression criteria defined and agreed before the start of the project. The funding in this call covers direct project costs with 2-tier funding pots, up to £1 million British sterling for product development projects, so TRL 4 to 5, or up to £300,000 British sterling for technical feasibility projects in TRL 3. Though as mentioned, we will consider late stage TRL 2 with reasonable evidence. I'll come back to discuss further detail about eligible costs and a brief summary of the terms and conditions in later slides. So as highlighted earlier, our intention is not just to fund, but to add additional wraparound support to enable the best chance of project success and to attract further investment following completion of the programme. The specific requirements will be bespoke and tailored depending on the needs of the applicant and the project, but the intention is to provide support in the form of support through application process. This could include one-to-one discussions and advice on developing the full application, as well as the catalyzer workshop, diagnostic development and disease area insight, advice and mentorship, providing access to a collaborative network of potential project delivery partners, to facilitate and support project delivery where applicable. So for example, development of statement of works or acting as a point of contact with delivery partners, to convene and facilitate scientific advisory boards and public and patient engagement panels, and help with business support. You can find out more on our website and the type of support we can offer. So I've taken you through what PACE will offer, but for innovators to maximise impact and reach their potential, it's essential that funded recipients and the PACE team work closely together. Therefore, we have outlined the following expectations of applicants and funded recipients. So we want you to be engaged in the process and work collaboratively with the PACE team in all aspects of project development and life cycle if successful. We expect that each applicant, if successful following the expression of interest, will attend or send a delegate to the Catalyzer workshop. This will be virtual to help in the development of the full application. Successfully funded projects to start within 3 months of the receipt of the award letter and be committed to the project length specified. Each applicant should ensure legal and ethical requirements are followed, for example, to conduct the project in accordance with the terms of the award agreement. We expect the applicant should highlight and raise any issues or concerns in a timely manner through the process and life cycle of the project. I will end my part of this webinar with a summary of the key terms and conditions of the funding programme. Please visit the PACE website, the address is at the bottom there, for the full details and access to the detailed document. Of note around intellectual property, ownership of results and revenue share, the funding on offer is non-dilutive and all background IP and any arising IP during the project lifecycle will remain with the recipient. There will also be no revenue share clauses. For the funding, awards will cover 100% of direct eligible costs up to £300,000 or £1 million limits with permitted expenditure. So permitted expenditure is defined expenditure which is reasonably incurred and properly evidenced and is directly related to and necessary for the delivery of the project. Typically, this will cover an appropriate percentage FTE of staff plus consumables directly related to funded work, access to specialist equipment and reagents, contract research organisation work, and reasonable travel and subsistence expenses. Receiving funding during the project journey will be linked to achieving agreed milestones, and standard payment terms will be quarterly in arrears. So any questions at this point, please add them to the question box, and we will answer as many as we can in the Q&A session later. Thank you very much for your attention, and I'll hand over to the next presentation.
Hello, and thank you for joining today. My name is Tanya Taylor. I'm a principal analyst at LifeArc and a member of the PACE team. I'll be following on from Pete's talk, showing you the core eligibility and scope criteria, and I'll be explaining more around the process and the selection criteria, as well as giving you some helpful resources for filling in the expression of interest form. This is a high-level overview of the process and timelines of this call with some key dates in there as well. As you know, the call is now open and you can submit your expressions of interest through the web form online. You should submit them by the 30th of October to be considered for this call. If you have any queries in the meantime, we've also got a web form that you can submit any questions you might have to, and we will get back to you. Following the expression of interest phase, the applications will be triaged by an internal expert panel that includes members from the PACE group, that includes MDC, LifeArc, as well as Innovate UK. And successful applicants will be informed that they've been selected by the end of December. So you should mark the 14th of January in your calendar, because we'll be having a workshop at that point to give you helpful tips and guidance on the full application process, as well as the next steps that are involved in that. So at that point, you will be entering the catalyzer that we previously mentioned, that wraparound support to really help you and ensure that your projects are successful. You'll be given an assigned R&D advisor at that point, and they will be your first point of contact, and they'll be guiding you through the process. The application deadlines will be around mid-April, and following that, the applications will be undergoing detailed review by external and internal KOLs, and patient representatives will be involved in the selection process as well. So we are intending to make a decision by the end of June, and you will be notified shortly thereafter if your project has been successful in this funding call. This will be followed by a round of refining and reworking project plans and milestones based on the feedback that we've received before milestones are agreed and the project award agreements are issued. And we're expecting that projects will go live after that within about 3 months. So this is an outline of the expression of interest form that you will find online. And I'll be explaining the section briefly now, but you will find more explanation of what should go into each of the sections in the online form as well. As a reminder, that needs to be submitted by the 30th of October, and you can access it directly here via the QR code. I should also note that while we'll treat all the information submitted confidentially, we won't officially have a confidentiality agreement in place at this point. So be mindful of that when you're putting in the information. And this form is really a short overview of the project. So you'll see we've got word limits associated with all of the sections. We'll be expecting a much more detailed view of the applications during the full application stage. So the first few sections are asking for a few more details for you and any co-applicants on the application. as well as a few short drop-down boxes to help us quickly assess and place the application. You'll then be asked for some free text information that includes a summary of the project. So you should outline the technology that you're looking to develop, how it fits within the current care pathway and addresses the clinical unmet need, and that should include a high-level target product profile. And I'll be explaining more what we mean by target product profiles in one of the later slides. We then have a few more extensive sections around the scientific rationale as well as the research plan. So for the scientific rationale, that should be any supporting evidence you have for your project, and really focus it on the key supporting evidence for this, the purpose of the expression of interest. So that could include any evidence you might have for how the biomarker links to clinical outcomes, any feasibility studies that you've done, or even surveys for user acceptance of the proposed technology and test. You will be able to upload 1 page of additional supporting data with your application form here. The research plan, again, it's high level at this stage, but we're really looking for the key objectives, outputs and deliverables, as well as milestones for the project. And we'll be working more closely with you during the full application stage to rework and refine this. This should also include information on you and any co-applicants and the expertise that you already have with respect to the successful delivery of this program, as well as any gaps you might have that PACE could help plug. And the final section relates to the translational plan. So this is really more forward-looking to what happens following the successful completion of this project. That could be any plans or ideas you have for further securing of funds, or any clinical trials that you have in mind for the regulatory approval, and ultimately how you see this test being implemented. It also includes considerations around IP, so either IP that you yourself might file, or IP that you need to have access to in order to successfully pursue this project. These are the key selection criteria for the projects. So based on the information that you will be providing us in the expression of interest form, we'll be looking for some key criteria to determine which project should be selected for the next stage. The first set of criteria relates more to the performance characteristics and technical details of your test. So that includes characteristics around the speed. So we're looking for rapid tests that can be implemented at the point of care. We're also looking for innovation, and that can mean both innovation in terms of completely novel technologies that you invented, or improvements on existing technologies. And here we're really looking for step changes in those technologies rather than incremental improvements. And the test should also be adequately accurate for the intended use. The next set of criteria relates more to the potential for patient impact. So here we're looking for evidence or a plan to obtain that evidence that will really demonstrate improved patient outcomes, the clinical outcome measures that could include reduced hospital stay, reduced mortality rates, and so forth. You should also be able to demonstrate that the diagnostic test that you're looking to develop is suitable within the targeted healthcare setting. And that includes considerations around the healthcare infrastructure, workflows, as well as any IT infrastructure that it needs to plug in for reporting purposes. And finally, the test should be affordable. So while we're not looking for tests for projects at the stage that we're at here to have fully fleshed out cost-benefit analyses, there needs to be a good potential for cost effectiveness in those proposals. I've spoken about target product profiles previously, and I will now explain a little bit more what we mean by those and why we think these are so important. So we really urge everyone, and that includes therapeutics developers as much as diagnostics developers, to have a target product profile really from early on in their development program, because that really sets the end goal, that sort of North Star that you want to reach to, and really helps align with the clinical need. It also helps you streamline your work plan to make sure you're on track along the way. It's a document that evolves throughout time as new evidence emerges, and we'll be working with you through the full application stage to refine these target product profiles. So at this stage, we're really looking at sort of a high-level outline to see that you've thought about where you ultimately want to get to. Some of the characteristics that you need to consider here pertain to both patient and clinical needs, as well as to the diagnostic characteristics of the test that you're developing. And that includes target analytes and sample types that you might use in your diagnostic. Who are the patients that would ultimately be benefiting from this test, and in what setting would it be used? So would it be, lower healthcare levels, or does it need to sit in a laboratory? What is the targeted diagnostic performance, so accuracy, that you're looking to obtain with this test? And how does it fit into the current infrastructure and diagnostic workflows? And finally, you should also consider the cost bracket that you're looking to attain for this to be implementable. I'd highlight again that we will prioritize solutions that are addressing indications associated with the highest AMR burdens and that are suitable for the point of care. And we've inserted here a few links to target product profiles that you can use as starting points or as examples that you might want to align against. That have been developed by the likes of WHO, CARB-X, or the CF-AMR Syndicate, and they're all developed for diagnostics for the use in bacterial infections. Here I'll outline a little bit more the terminology that we're using for the research plan. So what we mean by objectives are the research endpoints that are delivered by the end of a project, while the deliverables and outputs are concrete and quantifiable results that you attain during the course of the project. Work packages are defined strands of work that are part of the project plan, and they usually result in the delivery of an output. And while the milestones, they're really the key tools to help you measure success along the way. They should align to your target product profile, and they're critical go, no-go points at which you will decide whether to continue with the project or not. And they can incorporate results from multiple work packages. So we will have to agree on the milestones prior to award, and we'll be working with you in the second stage to work on those. I hope this has been helpful, and I've provided you a good overview of the call and the criteria, as well as some helpful guidance for how to fill in the expression of interest. You'll find all of this information alongside FAQs and our terms and conditions on the website. But if you have any further questions, you can either post them now during the question and answer session, or you can contact us via our website form. So with that, I thank you very much for your attention, and handing back to Bev for the questions and answers session. Thank you.
Many thanks, Clive, Pete and Tanya, for those presentations. So we end our presentations now during this webinar, and what we'll move to now is to our interactive question and answer session for the remaining time that we have together on the webinar. So yeah, many thanks to so many of you that are posting questions already. If any of you haven't already done so, please submit your questions in that Q&A box on your screen. And it's possible we may not manage to answer all the questions today, but we will post the answer to all the questions and any we don't address today after our webinar on our website on a FAQ page, alongside the slides and the video presentation. So I am now joined by several members of the PACE team to help answer the questions that you've posed to us. So all of the speakers that you've heard from already, also Dr. Phil Packer, who's the Senior Responsible Officer from the Innovate UK side, a whole host of members of the science team from PACE, and also finance representation as well. So we've got a great cross section of the team here today. So without further ado, I think I will start looking at the questions that you've posed for us. I think I'll try and group these. Probably the first place for us to start is there's a number of questions looking at the scope of the diagnostics call. And I guess maybe to kick things off, there's a couple there around why we're choosing to prioritise diagnostic solutions for UTI, lower respiratory tract infection and bloodstream infection and sepsis versus other indications. I wonder, Amelia, whether you would kick us off with an answer on that one, please.
Yeah, happy to. So whilst scoping out the priorities for this call, we've done a lot of diligence to understand the AMR diagnostics landscape, and we decided to prioritise these indications as they represent areas with the highest burden of AMR, and they're also indications for which PACE is supporting the development of novel antimicrobials.
Thanks very much. I guess an immediate follow-on to that, there is quite a specific question here around sepsis, and we will post more details on this on the website, but the question is around are diagnostics working on blood culture rather than blood sample acceptable, or are we only looking for diagnostics that do not require blood culture? I think that might be one that's worth us picking up now. Pete, can I come to you for that one, please?
Yeah, sure. So I think there'll be more details actually when we kind of put the examples and the preferred characteristics out with the slide deck and things going forwards. But really, I guess what we're trying to say is we're not ruling things out, but what we are looking for is something that speeds up that kind of process, particularly for kind of blood culture as that's the time-consuming step. So if there is a replacement or something that generates results faster than kind of standard blood culture methods, that's really kind of where we're getting to. Or perhaps things that can go straight from a sample, or at least without a separate kind of step or extensive kind of sample prep. So yeah, a bit of room for maneuver in that space, but that kind of idea. And yeah, please do have a look at the examples when we share those as well.
That's great. Thanks, Pete. And I guess the other questions related around scope as well as disease indication, there's a few questions around the pathogens that are in or out of scope. Do you want to follow on that one as well, Pete? Yeah,
sure. So as we've said, we're looking for projects to kind of develop rapid diagnostics for UTI, LRTI and BSI. And really, I guess we want to prioritise proposals that address the key pathogens associated with those. So particularly in relation to AMR. So, you will have seen the kind of various articles in the Lancet in terms of, burden of disease, etc. And that's a reasonable good point for kind of guidance. But, for example, if we had a UTI diagnostic, if it wasn't detecting E. coli or Klebsiella, then that maybe you're not addressing the kind of the key pathogens there. I guess for any proposal, but particularly kind of perhaps more targeted diagnostics, the positioning, the use case, kind of how it would have potential clinical impact, and the things that kind of Tanya's talked about. That's they're the key kind of considerations and need to be kind of clearly explained. And just to reiterate, we're not looking for diagnostics, looking at pathogen ID alone as well. So we want additional information than that.
Great, thank you very much. I think that's all the core scope questions around the kind of disease indication and pathogen. We've had a couple of very closely related questions here around what we mean when we say tests require central lab infrastructure and that is out of scope. Paul, could I come to you for that one, please, just to explain and bring that to life a bit more? Thank you.
Sure, thanks, Beth. Thank you for the question. The answer is pretty straightforward, actually. For this funding call, we're focusing on point-of-care technologies in the healthcare settings, levels 1 and 2, right? So the reason we're doing that is because that's where we think that's where the greatest unmet need is after our diligence. So we want to position new diagnostics, improved diagnostics, even repurposed diagnostics in those settings, because that's where we think that will make the greatest impact on AMR from the funding available to us. We do recognise that there are unmet needs in level 3, but that's not a focus of this call. Thanks.
Thanks, Paul. Yeah, I think I might move to a new area now of questions that are coming in. It's more, these questions are more related to the kind of support that projects might be able to expect and what people need to be able to put in their application. So there's one here which is, do project teams need to have the capabilities, all of the capabilities, tools and facilities to deliver the project in-house in order to apply? Pete, could I ask you to pick that one up, please?
Yeah, sure. So I guess the short answer is no, but we do ask that applicants, kind of articulate a clear plan as to what is needed. So, what capabilities, tools, facilities, and as part of a kind of, overarching research plan. But that could come through collaboration with partners or outsourcing defined parts of the research plan. So CROs, for example. We want people to show, value for money in that kind of outsourced work. But yeah, you don't have to do everything yourself. Obviously, smaller companies can't always do that, but we need to be realistic about that.
Yeah, and on that, I wonder, Adriana, if I could ask you, Adriana has spent a long time over the past 12 months working to convene a delivery partner network who is looking to work very closely with PACE to help support PACE supported projects. So Adriana, can I allow you to say a bit more about that, please?
Yes, of course, Bev. So yes, we have set up already a delivery partner network, so a bunch of CROs that we selected, and we selected because they cover capabilities in the drug discoveries to help PACE and support the PACE program, and we will help you getting access to this network of partners. The idea about the PACE Delivery Partner Network is to develop it and to evolve it with the PACE program. And so because of that, at the moment, we are engaging with new CROs and we want to add them in terms of covering diagnostic expertise and capabilities, which will include things like product development, manufacturing, lateral flow development, regulatory, so a little bit of everything really. And this will keep evolving, as I said, during PACE. So we will always have new capabilities when necessary.
Thanks very much, Adriana. I think I'm going to switch gear again. There's quite a few questions that are more focused around the application process. So I think it'd be useful to take a few of those. One of them is around whether applicants can be listed across multiple applications. Tanya, can I come to you for these scope process questions, please?
Yeah, sure. Happy to answer that. So yeah, absolutely. You can submit multiple applications, or you can be listed as lead applicant or co-applicant on multiple applications, so long as they're separate, distinct and unrelated. So they're two separate projects. I think there are some related questions there as well on the, can I collaborate with someone abroad? Does all of the work need to be conducted in the UK? The answer is no. No, it doesn't all have to be in the UK. So this is a global call. It can pretty much come from anywhere in the world, and we're happy to support cross-national collaborations as well.
Great, thank you. And then a question about the process. And so after people have submitted the expressions of interest, will feedback be provided at that stage? Tanya, could I come to you again, please, for that one?
Yeah, happy to answer that. Yes, so the aim is to provide feedback or let you know the outcome of our decision as soon as possible after we've reached that decision. So you should expect a notice by the end of this year in terms of whether you've made it to the next stage. And we're expecting to roughly take, 15 or so through to the next stage to give you sort of a guidance on that. The feedback at this stage, given the volume of applications we're expecting, will be high level, so we can't provide detailed feedback to everyone.
Thanks. Another related one around eligibility to the process is an academic partner necessary? Can an SME apply on its own? Tanya?
Yeah, that's fine. So you don't need to have an academic partner. We are happy to support SMEs as well as academics. You can have industry collaborators on that. The only thing that we ask in that case is that the industry collaborator carry their own costs.
Thanks. There was a related question around how we define an SME, we will provide this. We have a table that we can share, which we will do on the website via the FAQs. But in essence, there are qualifying criteria based around turnover, balance sheet, total and number of employees that we use to define whether an organisation is an SME. We will share that information after the call. I think there's a few questions more closely linked to finances and funding. I guess maybe one general one to start with is what expenses are covered? Are salary costs eligible? Stacey, please can I come to you for these finance questions?
Of course, thanks, Bev. So awards will cover 100% of direct costs. Obviously, they have to be sort of eligible, and we'll provide some permitted cost guidance that will be made available to you. But essentially, costs, sort of examples of costs that you can claim would be an appropriate FTE for staff time, any consumables directly related to the funded work, access to specialist equipment, CRO work. I think there was a bit of a question around, can we use subcontracting from another country? The answer is yes. If you are sort of UK based and wanting to use a company abroad, you just have to provide a justification as to why is there not an alternative in the UK available, and you know it has to be necessary for the project. And so as long as you put that in your application or your expression of interest, then that's absolutely fine. And I think there was a couple of questions as well, Bev, around sort of payment schedules and things, so I'll probably just address that as well, if that's all right.
Great.
So the funding will be paid quarterly in arrears based on your actual costs that have been incurred. And it will also be paid on the basis that the cash has actually left your bank. So it has to be incurred and defrayed for us to pay it. And I think, as alluded to earlier, there will be milestones kind of built into your project plan. So as long as everything is on track and we're meeting those milestones, then we'll continue to kind of fund those quarterly claims. Thanks, Bev.
Thanks very much, Stacey. I think we have answered this. There's one question here, someone double checking university overheads would not be considered eligible.
Yeah, so university overheads or any overheads are not eligible for this funding call. It's 100% of direct costs only.
Yeah, and there's a few questions I'm not sure that I've addressed here that are around eligibility, about where applicants are based. So yeah, just to reiterate, this is a global call. We absolutely welcome applicants from any geography, UK, non-UK, everywhere in the world. It is very much an international call. And that includes for, yeah, the lead applicant. So the lead applicant can be based anywhere, any geography. And there is a question sort of financy. If a company has not received any previous funding in the past, will that have an impact on their application? I don't know if anyone on the panel wants to take that, or I can have a go. Does anyone want to jump in? I can take that. I guess what we are looking here is to look at the projects on their own merit. We're looking to find those most innovative projects, those projects with the most transformative potential, and that's what we're using for that assessment process. And we will do that in partnership with a group of independent science experts to help us do that. Yeah, I'm jumping around a little bit now as new questions come in. There's one here around if we want to benefit from the PACE network of product development, manufacturing as a project partner, shall we articulate this in the expression of interest? I think, does anyone from the panel want to jump in on that one?
I can take that. So there is a specific session in the expression of interest form where we ask for sort of gaps and needs that you have that we could support you on. So you can put it in there, yes.
Perfect. And I think the other thing to say is that as we progress through the application process, those people that are invited to then submit full applications, there will be a catalyzer program during which we would look, the R&D advisors on PACE would be looking to help support any connections that need to be made to help during that stage. I'm just scanning through to see if I've missed any questions, which I may have done. And so, yeah, I will reiterate, we will go through all of these questions and make sure we do have a proper response in the FAQs to them all, which we will post. I don't see any others. Unless anyone from the panel wants to jump in on anything that I've missed.
I think that there was just one thing I picked up on. I think there's some questions related to sort of One Health approaches, looking at sort of environmental reservoirs of bacteria. So that's currently not in scope. We're only looking at human targeted diagnostics in the 3 indications that we've listed.
Thank you, Tanya. Okay, fantastic. So it's been super to have so many of you on the call with us today. I think that ends our second PACE webinar and our Q&A session. Hopefully we've provided you with a lot of the information that will help. Many thanks for your interest in the PACE funding round. We will share an updated FAQs. We will also make the recording available, the slides available. But please do contact us via the website should you have further questions, and we will endeavour to answer them. I should add that the call for expressions of interest is open now. It is open until 23:59 GMT on the 30th of October. The link to the submissions portal is live on the website now. So yeah, we really are looking forward to receiving your applications. And I think that's us. So thank you so much for joining, and goodbye.